Newton, please go ahead.
Okay. Good morning for all. Welcome to the presentation of PhD thesis of Caetano Sabino. I thank to the professor and friends and relatives of Caetano for honoring the thesis. Caetano is a PhD degree student in the School of Pharmacy at the University of Sao Paulo in Brazil, at the Department of Clinical Analysis. The PhD work is entitled Inativação fotodinâmica antimicrobiana mediada por azul de metileno. Análisis de cinética de inativación y mecanismos bioquímicos. Antimicrobial photodynamic inactivation mediated by methylene blue. Analysis of inactivation kinetics and biochemical mechanisms. I am the advisor. My name is Nilton Linkopan. I'm a professor in the Department of Microbiology in the Institute of Microbiology in the University of Sao Paulo. The co-supervisor is Professor Marta Ribeiro from IPEN. And informally, another co-supervisor is Professor Mauricio Baptista from the USP. The panel of evaluation is made up of Professors Michael Richard Hamblin from Wellman Center for Photomedicine, Massachusetts, Boston, United States. Professor Matt Wainwright, Professor of Antimicrobial Chemotherapy, Pharmacy and Biomolecular Science in the Liverpool John Moores University in the UK. And Professor Jorge Luis Melo Sampaio from our institution, the School of Pharmacy of the USP. Thank you all evaluators for your participation. Let me start by giving you some background information. Okay, time starting 9 p.m. and ending near to 1 p.m. Summarizing 3 or 4 hours. I believe that we could finish the process before. Caetano, firstly, you have 30 minutes for perform your presentation, and then after presentation, each member of the evaluation panel will have 30 minutes for question and answers. Finally, only the member meeting to give the final opinion, and in another room or after disconnection of the public and the evaluated student. And in the final part, the decision will be publicly read. Caetano, are you ready?
Yep.
Start working.
Thank you very much. Thank you very much, Newton, for the kind introduction. Thank you for helping me for all this time. It's been 5 years already. Martha as well. It's been 10 years that we have been working together. Mike Hamblin. It's been almost 10 years since I've last been there to Boston with him. Mark Wainwright's a very good teacher, professor, and friend as well. Thank you very much also Mauricio, who might be watching us from far away right now. And Professor Vanderlei Banhato and Professor Georgia, thank you very much as well for being here. So I shall begin, and I'll share my screen right now. I hope you can see it properly by now. Is it okay?
Yes. Yeah.
All right.
Yeah.
So let's begin. So the big problem that we had before COVID-19 was superbacteria. They were in fact the #1 priority for the WHO. We have those crazy numbers down here that were pretty much doubtful before the COVID pandemics because people didn't think how bad this could get. But with a bacterial pandemic, which is much more serious if we lose the activity of all the antibiotics that we have in the market right now and that are being developed as well, the WHO estimates that we can go up to 10 million deaths per year by the year of 2050. It's like 4 times more than what COVID did to us so far. So it's going to be pretty serious. And it's not just one strain, one species, it's several of those. So it's very complicated Also, the World Bank has estimated that it could cost about $6.1 trillion per year on excess in healthcare. That value is pretty much the sum of Brazilian and Great Britain's GDP to get this kind of number. So it's really serious, and we really need to do something by it now. This is a declaration from the Director General from the WHO, saying that if we don't do anything and we actually get to a post-antibiotic era, a child scratched knee could kill again, as in the Dark Ages, or even worse, we may have to do amputations due to those simple infections that used to be not a big deal for us today. So thinking of that, the WHO also had a campaign that it supports every year, a World Antibiotic Awareness Week. The first one that I have registration here is from 2015, which is just before I began my PhD. So our focus was to face this problem. And guys here working on antimicrobial PDT really are tired to know that this is the main cause that we're developing this technique. We have a very robust technique that can face all those drug-resistant bugs. And so far, due to the misuse of antimicrobial chemotherapy, we have been selecting all sorts of drug-resistant bacteria and fungi and protozoa. And the resistance level is getting quite alarming right now already. Big problem is that bacteria can share some drug-resistant genes in between each other. There are a few mechanisms of that. This is just conjugation through a sex pilus, so they can even make a bacterial sex to share drug-resistant genes. And what we do here, and pretty much everywhere in the world, we throw everything together. Here in São Paulo, this is a really bad smelly river that we have, which is Rio Tietê. Mark Wainwright and Hambling already saw this kind of thing. George and Newton and Martha are here looking at it every day. And so we mix all the garbage and sewage from hospitals, residences, industries, and everything together so that the bacteria can share their drug-resistant genes in those things. And we even have a Brazilian variation of a metallo-beta-lactamase. There is a carbapenemase, which is SPM1, and it was found in this very river. Okay, so this is the kind of things that we have been doing so far. And we urgently need something else, some innovative, alternative antimicrobial method that is not susceptible to drug resistance. And antimicrobial photodynamic therapy is a very good shot for that. It combines, pretty much consists of the combination of a light-sensitive drug with light of resonant wavelength, with this absorption of this drug, of this light-sensitive drug. And with this kind of combination, we can pretty much kill any type of microorganism. Doesn't matter if it's a bacteria, fungi, protozoa, or viruses. We can generate sufficient oxidative stress through this technique to kill any of those without any kind of selection. It's very simple to be performed. This is an example of treating for some diabetic foot, which is a project that I like very much from our great partner, who is Professor João Paulo Tardivo. We are also scheduled to have a large-scale clinical trial, which will be multi-centric here in Brazil. It was supposed to begin on March of last year, but the pandemic took off, and we are still waiting for it to get slower and dimmer so we can begin our tests. But pretty much we just need to spray the photosensitizer, the photosensitive drug that I've been talking about, and then activate it with light. It can be either laser or LEDs, because monochromatic light is usually the most efficient way to excite this photosensitizer. And it doesn't even need a surgery room. It can be performed any ambulatory or some simple clinics or even at the patient's home. So it's very convenient to work like that. And we can pretty much apply that for many, if not nearly all, kinds of localized infections that we might have in humans and also in animals. A few years ago, we even issued a book on photodynamic therapy for veterinary sciences. Was actually the first book in this specific area. Mike Hamblin and Professor Martha are both here representing some of the folks who participated in the craft of this book. So the mechanisms of photodynamic therapy are quite interesting. They involve some quantum mechanics to describe what is actually going on when a photosensitizer, symbolized by PS here, absorbs some light, symbolized by H&E here. It goes to an excited state in a singlet excited state, and it can go through an intersystem crossing to go to an excited triplet state. And that state, it has mostly two paths to go through. Here I only exemplified through direct interaction with oxygen, with triplet oxygen, but it can also interact with some biomolecules instead of triplet oxygen. But the special thing that this excited photosensitizer can do is to even either donate an electron to this triplet oxygen. Then it becomes a radical, a superoxide radical. And this superoxide radical can also be further reduced to hydrogen peroxide. And hydrogen peroxide can undergo some phantom reactions and release hydroxide radicals. And this stuff is really, really reactive. And it can interact and damage pretty much all kinds of biological molecules present within a cell. This is the type 1 kind of reactions. We also have the type 2 kind of reactions. And in this special type of reaction, we actually have an energy transfer, an energy donation from the excited photosensitizer to a triplet state oxygen. And then we have a ground state photosensitizer and an excited singlet state oxygen, which is quite reactive as well. And this thing can also react with many kinds of biomolecules present in the cell. And it can also even be reduced with one electron and become a superoxide anion radical and undergo the other reactions from type 1 reactions. So it has these two paths to go. And both of them culminate in the generation of highly toxic and reactive compounds within a microbial cell. Interesting thing is that if we go through the type 1 reactions that generate radicals, we have quite a few cellular responses that can protect it against these molecules. And those protections are even based with enzymes that are detoxifying, let's say, the cell from these reactive oxygen species. First one that can interact with superoxide is the superoxide dismutase that generates hydrogen peroxide. And hydrogen peroxide can interact either with catalase, as this example here, but also with some glutathione reductase and so on. But if we have this phantom reaction going with hydrogen peroxide, we generate some hydroxy radicals that don't have any kind of enzymatic defense against it. Alternatively, if we go through the type 2 reactions, we directly generate singlet oxygen that doesn't have any kind of enzymatic defenses. So we only have quenchers, scavengers, and so on to defend the cell from this kind of compound. And it can react with pretty much any type of molecules, of biomolecules, but it's not as reactive as the hydroxyl radical, okay? But both of them can lead to extensive damage in the cell, causing protein denaturation, cross-linking, aggregation, lipid peroxidation, can cause strand breaks in DNA, can cause formation of oxidized bases such as 8-oxo guanine and and all sorts of damage in the cell that can lead it to cell death directly. If we are talking about microorganisms, there's no such a thing as apoptosis, but so they all die by kind of necrosis. But we're going to see some of the mechanisms and how we can interpret this kind of death with in this presentation. Okay. So the main objectives of this this thesis has been divided into 4. And those are configured as scientific articles. It's a format of thesis that we have here in our department. We can use 2 or more papers that we published, or they are in the process of publishment to assemble our thesis. So I got the 4 major papers that make a nice story here to compose this thesis. And the first one is about a mathematical model to allow precise analysis of photoinactivation kinetics, because so far it's been quite complicated to get to further insights based on the inactivation kinetics of bacteria and microorganisms in general. Second paper is an extensive analysis of APDT effectiveness against all sorts of global priority drug-resistant pathogens. We also have some fungi there, as well as all those top priority bacteria that WHO has established. Number 3 is about the inhibition of ß-lactamases by APDT and also antimicrobial blue light. That's very interesting and very powerful insight from our mechanisms of action. And the 4th one is the only one that is not published yet, and I really wanted to focus our discussions after my presentation on this last one, because we have the chance to improve it a lot yet, and I really want your feedback on that. And in that last paper, we are going to try to establish the actual biochemical mechanisms of APDT. So we know that there is all this mess going on inside the cell, but we want to know what actually causes the cell death. What is the first component that goes off and the cell dies, is unrepairable. So the first one here is about this mathematical method that we actually adapted from other applications, even from UV germicidal radiation. They sometimes use this method. It's pretty much when we do all sorts of inactivation kinetics analysis for APDT or antimicrobial blue light or even UVGI, we have some very messy data, because we need to analyze several data points of light doses. In the case of PDT, we also have to analyze some variations in concentration of the photosensitizer. So comparing just point by point, doing paired t-tests and so on, it's not very good, and it gets quite messy when we have a large data set. Just linking those data, it's also easier to visualize them, but doesn't bring any insights to us, doesn't bring any further data that we can use and explore what is going on there. So we need some reliable and precise method to analyze this kinetics here. And then we can extend this model to see what happens in very large doses that we were not able to test. We can make all sorts of simulations with that. So it's very interesting to have something like it. And we did it. We actually adapted a power law, which is correspondent to what the Weibull method uses. We just made some adaptations to make it clearer and more contextualized in our specific application of photoinactivation. It's a quite simple equation there, which you can see down here. It's actually my handwritten description of the formula, which is pretty much just the log reduction of our inoculum that is equal to a dose that can either be in radiant exposure, such as joule per square centimeter. It can be number of absorbed photons, it can be time, it can be whatever kind of unit you'd like to describe this dosimetry of light, because they're all linearly correlated. So that's our main variable there, and that gets divided by a constant, which is the little dose for 90%. Okay, this information is quite interesting for us already, and all of that is on the power of T. T is a tolerance factor that will tell us if the inactivation curve has a concavity upwards or downwards, or it's just a straight line. So some simple tool that physicists use to analyze if a phenomena is describable by a power law is just to apply a double log scale on both axes, on y and x axis, to see if those data linearize in a way that we can fit a linear curve here to those data. And indeed, it actually did. All those data, all the data that I actually ever tried so far that has some good quality of data, always fitted to this model. And we can even see down here the residuals of those fits, and they're quite random there. So it tells us that it's a quite good fit without much influence of outliers and so on. And there you go. So here the top graphs are already being used, the power law function, to fit those data. You can see that we can extend the fitting line, so we can get insights to higher doses. We can even know now what is the lethal dose for 100% of inactivation of some bug. That's something that we could not do experimentally so far. We can also get those fitted curves and apply some derivative analysis, so we can see how the inactivation kinetics rate changes over time. And you can see here that there are some microorganisms, especially for methylene blue APDT, that begin in a much lower inactivation kinetics and speed up over time, as well as there are some other bugs that start very fast in their inactivation kinetics, but then stabilizes and get under a mostly constant trend. For blue light, it's always the same kind of behavior. They actually begin slowly and then speeds up the inactivation kinetics. So that's quite interesting, and that's one of the insights that we can get from this fitting. You can see down here on the bottom left side that all the fittings were really good. All adjusted R-square values were above 0.95, which is really good. Usually anything above 0.8 we consider good. And here are some of are the constants that we can directly extract from that fitting, which is the tolerance factor T, that I named that because it tells us if a microorganism has some more tolerance to photodynamic inactivation or photoinactivation in the beginning of the process or at the end of the process. If the tolerance factor is greater than 1, the concavity of the curve is pretty much like this blue light. So it has a shoulder, it has the concavity facing downwards, okay? If the T factor is equal to 1, these curves become a straight line, pretty much. Doesn't change its trends. Might be something close to what these Pseudomonas inactivation kinetics trend is shown here on the methylene blue APDT graph. And if the tolerance factor is lower than 1, then we have the other way around. We have like this E. coli and Staph aureus trend here for APDT, which is with the concavity facing upwards. So they are actually very sensitive to inactivation in the beginning of the process, and over time they get a little more tolerant. And the lethal dose for 90%, that's some very interesting data to have in experimental biology when we want to either kill some cancer cells or microbial cells. Knowing the lethal dose for 90% is something that is very interesting when we are working just with a chemical compound, it's quite easy to get there. But when we are working with this mixed techniques such as photoinactivation, it's not so straightforward to have it, but now we have a very easy method to calculate that, okay? And we can even use some little algebra and figure out a formula to describe any level lethal dose. So we just need to input those constants that we extracted from the fittings, which are lethal dose for 90%, and the tolerance factor. And the I here is respective to the level of inactivation that you want to have. Here I already transformed to percentage, but we can transform back to logs. Okay, so if we wanted to have a 99.9% or 3 logs or 4 logs or 5 logs, we just need to change this factor I, and then we can calculate what is the lethal dose. And you can see here from the standard deviations that we have in these graphs that the method is quite precise to figure out what is the lethal dose for those bugs. So it's very interesting, a very simple and powerful tool that it will be a fundamental tool for the rest of the studies that we did in this thesis. So the next one is the photoinactivation using methylene blue mediated APDT against lots of strains from global priority multi-drug resistant pathogens, according to the WHO. So what we did is to get a wide variety of drug-resistant strains and compare them to their ATCC drug-sensitive controls, okay? And pretty much within this collection here, we have nearly all types of drug resistance in bacteria that we can ever find. So if we have some chance of cross-resistance in between antimicrobial PDT and antimicrobial chemotherapy, it should show up something here, okay? So these are the kinds of antibiograms that we have for those strains. You can see that it's quite bad. So the drug-sensitive ones are shown by this blue arrow. The MDR, the multi-drug resistant, those that are resistant to more than 2 classes of antibiotics are indicated here through this red arrow. And the XDR, the extensively drug-resistant pathogens or strains, they are indicated here by this purple arrow. And those are the microorganisms that are sensitive to less than 2 classes of antibiotics. So we have some quite bad collection here, and we tested everything. So we have like around 30 strands from about 10 species of microorganisms, and now we can use all this mathematical model to fit those data and have a clear comparison between those. And we can even look that we have 2 different sorts of sensitivity scale of those bugs. There are some microorganisms that respond within less than 5 joules per square centimeter of APDT dose already and exposure to get to more than 4 log inactivation, but there are other species that actually need about 25 to 30 joules per square centimeter to be inactivated to more than 4 logs, okay? And one interesting thing that we have here, especially for the APDT community, is that it's quite popular to have people declaring that gram-positives are more sensitive than gram-negatives, that are more sensitive than fungi. And this is not absolutely true. Nature is highly complex. And we can see here in these graphs, for example, that Enterococcus faecium, this VRE strain, and the ATCC drug-sensitive control, they're both more tolerant to APDT than, for example, E. coli, which is a gram-negative. So yeah, Enterococci are actually more drug-resistant, more APDT tolerant than a gram-negative bug. At the same time, we can also see here that Klebsiella pneumoniae, it's actually more tolerant to APDT than Cryptococcus neoformans, which is a yeast. So we also have gram-negative bacteria being more tolerant than yeast. So we have a more complex situation. I'd love to have a chart on what actually causes each of those species to have this particular inactivation kinetics, but it's a highly complex and complicated situation that we need to still figure out. If you have some insights on how to get that, I'd love to hear it afterwards. So then, with our analysis, we can also calculate the t-values for all those species. We actually didn't have any significant difference in between different strains of the same species, but when we compare different species among each other, they get quite different. They get some relevant differences in between each other. So PDT is pretty much species specific, okay? And we can see here that, for example, Staph aureus, which is generally taken as a very sensitive bug, very sensitive bacteria to APDT, it actually has a very low t-value. So indeed, in the beginning of the photoinactivation process, it might get some very strong inactivation, but afterwards, there are some persistent cells that will remain harder and harder to kill. On the other hand, if you get, for example, Klebsiella pneumoniae, which is the highest one here, it's quite tough to begin to inactivate this bacteria. But after it takes off, it gets quite faster and faster as being inactivated. And we can see that much more clearly here when we plot all the lethal doses for 90, 99.9, and 99.999, respectively 1, 3, and 5 log reduction for each one of those species. And you can see that in the beginning of the process, for the lethal dose of 90%, we have a very big variation of doses. They can reach up to 100 times variation. And as we go on and on over the inactivation process and reach here the 5 log level, we can see that the differences are no longer that wide. Okay, so we have up to about 10 times or 1 order of magnitude. So we lost another order of magnitude of difference from LD90 to LD99.999. And with this kind of analysis, we can actually set a minimum dose that covers all the different species that we have studied here. So for example, if we established that we needed to have 30 joules per square centimeter, we would be able to inactivate more than 5 logs of any type of bacteria or yeast that we tested here in this experiment. And that's quite interesting to have some robust protocols of photoinactivation. Okay, so then the third paper that we have here to present is about inhibiting ß-lactamases, which are probably the most challenging thing in microbial drug resistance that we have. So they are a major challenge, and we already showed that we are able to inactivate all types of drug-resistant bacteria, including those that have ß-lactamases. And that doesn't make any change on their sensitivity to APDT. But one interesting question is that, can we also inhibit those ß-lactamases that might be still active inside those cells? Because they're not dependent on ATP, okay? So even if we extract those ß-lactamases from the cell and just work with it in solution, they still can cleave the beta-lactams that they're effective against. So here in this study, we tested methylene blue mediated APDT as well as antimicrobial blue light to inactivate some pretty bad Klebsiella pneumoniae strains that contain at least 3 Ambler classes of ß-lactamases. And then we fitted our curve there as well. And now we can compare directly under the same level of inactivation for each one of those strains. So what we did here in this study is to analyze those very strains after exposure to APDT or antimicrobial blue light, and get the only study them their activity of their ß-lactamase on the exact dose that we need for 3 log of inactivation. And that's what we got. So yeah, we can, both techniques can inhibit at least class A, B, and D of Ambler classification ß-lactamases. These specific strains had IMP, SHV, CTXM, KPC, and OXA strains. And we could inactivate or inhibit all those enzymes, those ß-lactamase enzymes, just in the same way, either for APDT as well as for antimicrobial light. And we also even have some controls here with methylene blue only and red light only. And of course, it didn't do anything. We need to combine them to have any types of result. So yeah, we can inactivate drug-resistant microorganisms and also inactivate what makes them drug-resistant. So yeah, science for that. That's pretty cool. And then we have our last paper, which is the one that we want to have a special attention here. It's yet being assembled. So the big question is, okay, so we have damages everywhere in the cell. What actually makes them die? What actually is the major target that we have in a cell, in a microbial cell, that is the most sensitive one or the most critical one to sustain the life of that cell? How can we actually even optimize that method? So in this study here, we use a methylene blue at 100 micromolar, as well as all the other studies that I presented to you. We use a 662 LED at 100 milliwatts per square centimeter, just like all the other studies. We can even calculate how many photos are being absorbed per second by this combination of red LED and methylene blue. We can get to this very nice value here, which is quite interesting to see that we have 2.62 times 10 to the 22nd power of photos being absorbed per second in this solution. It's just 1 milliliter of methylene blue being irradiated by 1 LED at 100 milliwatts per square centimeter. And each methylene blue molecule, since we know how many of those are in there, we know that they have about 50% of quantum yield of singlet oxygen. And we can calculate using some of these methods down here, this mathematical model down here that we have on photo absorption rate. We can actually calculate that each methylene blue molecule can yield up to 2.1 times 10 to the 5th molecules of single oxygen per second. So that's a very strong oxidative power that we have there, with only talking about singlet oxygen generation. We also have those type 1 reactive oxygen species being generated there. And we actually even measured that the phosphorescence of singlet oxygen, which is an excited species and emit this 1270 photons of phosphorescence here with this nice curve. We did these measurements with Professor Mauricio's team. We can even measure the lifetime decay of this singlet oxygen being formed there in this medium. We also try to mix it with 10 to the 8th CFU per ml of Klebsiella pneumoniae to see if we have any quenching of this signal, but we did not. I suppose that is because we in our solution we had so much more methylene blue just solved in solution, much more than within the Klebsiella pneumoniae cells. So then it just doesn't give some changes in the signal that are enough for us to quantify here in this macroscopic kind of analysis. So our first step was to use some confocal fluorescence lifetime imaging microscopy to see where this methylene blue ends inside a cell. We have some publications from this group, the Swiss group that is composed by Nogueira and Gonzalez, showing that they did all theoretically and in computer simulations, showing that methylene blue can actually intercalate very well in DNA strands. And when it does, it gets some metastable disposition inside this ladder of DNA, and actually increases its stability in the excited state for singlet and also the triplet states of this photosensitized methylene blue. So since we have a more stable state of singlet methylene blue, excited singlet methylene blue, we have a change in the fluorescence lifetime. And then since we also have the stabilization of the excited triplet state, we also have, according to them, an enhanced yield of singlet oxygen. But let's just look at this fluorescence lifetime imaging. We actually use it candid outcomes here just as a reference of position of DNA material inside a cell, since it's a compartmentalized cell. So, and here we can see really well, here it's just methylene blue stain, okay? We're just looking at methylene blue here. And in the red signal, we can see everything that is inside the cell interacting with its intracellular components, mostly proteins there. But when it gets to the nuclei and mitochondria, it triples its lifetime with those DNA-rich components. So then we have this merged image that shows very much that we have two distinct populations of methylene blue. One that is within the cytoplasm and the other that is inside the nuclei and mitochondria. But our target of study here is Klebsiella pneumoniae, which is a not compartmentalized cell. It's a bacterium, gram-negative one. And we can see here that we also have both signals, that one of 0.6 nanoseconds and the other one of 1.8 nanosecond. And when we merge those images, we can see that we have some distribution of that, even though our spatial resolution for this level of precision that we have here is not so great. So it's a very pixelated image, but we can see it clearly that methylene blue is inside those cells and interacting with DNA and proteins. So then we began working with some photoinactivation of this Klebsiella pneumoniae. That most of the studies we did with these ATCC 700603 strain, which is a drug-sensitive, relatively drug-sensitive strain, which is safer to work. But in some studies, we had to do some testing with other bacteria or other strains of Klebsiella pneumoniae that showed some drug resistance for some comparison. You're going to see it later. But just keep in mind, their inactivation kinetics is pretty similar here. We don't have any significant difference. So doses that is valid to one will be valid to the other. And here, under these nanoscopy images, we can see that the control cells, just talking about the techniques, here on the upper side, we have just transmission electron microscopy, which is a classic technique. But the one underneath it is atomic force microscopy coupled to infrared spectroscopy. It's a very nice technique that can enhance the spatial resolution of infrared spectroscopy by more than 1,000 times, breaking the diffraction limit of imaging. There's some trickery of the atomic force microscopy. But what is interesting here is that we can actually establish a specific wavelength or wave number that we want to scan this sample. In this case was the amide-1 absorption band, which shows the protein content of the cell. So the control cells here, either on the transmission electron microscopy or the atomic force microscopy, they're quite evenly distributed in respect to their intracellular components. The membranes are just fine and so on. But when we expose them to APDT, and that's a very high dose of APDT, here we use it 100 joules per square centimeter, and you can see on the graph on the left that we can get more than the 4 logs of inactivation only at 25 joules per square centimeter. So that's a fourth of the dose that we use it for these images here. We really wanted to take the dose very high because we wanted to see the ultimate kind of damages that we can input to this cell. The only thing that we could see here in these doses is that the protein, most of the intracellular components, they just get aggregated over the periphery of the cell. And the DNA strands here in the middle, they just look like a little more naked and spaced in relation to the rest of the cellular components here. And another important point is that we don't ever see membrane rupture. We don't see any ghost cells. We just see them quite deformed like this. Maybe the the inner membrane detaches from the outer membrane a little bit according to these images, but we don't see them rupture, okay? And the same thing can be visualized here in the atomic force microscopy coupled to infrared spectroscopy. So it's a very interesting beginning point. So membrane damage might not be the most important thing there. And then we tested DNA damage and also protein damage and also membrane permeability here in these graphs. So let's begin here on the right side on this pulsed field gel electrophoresis image here, which is the top right side one, where we have the entire chromosomal DNA just split in a few segments due to the use of a restriction enzyme. So we can have this beautiful ladder pattern here with all those specific bands according to their size. All the controls didn't show any difference in those bands configuration. And when we begin exposing those cells to APDT, we see that at 2.5 and 5 joules per square centimeter, we have pretty much no changes in those band patterns. But when we reach 10 joules per square centimeter, we already have this mirror band being formed down here, which shows that DNA strands are being spliced into very tiny pieces. They are random, nearly random in size. If we go to 50 joules per square centimeter, we pretty much almost have nothing here. The DNA pieces are so small that they're not kept inside this gel, only at the first well up there, where we have the largest pieces of DNA. But if we go to 100 joules per square centimeter, we don't even have those DNA strands being accumulated in the first well up there, in the first band up there, I'm sorry. So yeah, we're having some DNA damage, but it's not like really fast. It kind of happened lately within the inactivation process. They're not beginning right after the first doses to show some damage. So they might be kept safe for a while before they begin to get damaged. Below here on these other gels that we have, those are PCR gels. And we kind of did this technique with the thinking that if we damage a segment of a DNA that's within that amplicon that we are targeting for, the DNA polymerase enzyme may not be able to pass there, so we won't be able to amplify that segment that we're interested in. So we use it to drug-resistant genes, but it's not just about the genes that we wanted to look. It's about the size of the amplicon. So this black KPC2, it's about 800 base pairs in length, and this black IMP1 gene is about 400 base pairs in length. So one is the double of the size of the other, okay? And we can also see that their sensitivity to APDT inhibition is pretty much one is the double of the other. One begins to get some inhibition by 20 joules per square centimeter, and the other one begins to get at 40 joules per square centimeter. So the size of the target, the size of what actually is the length of the DNA sequence that we are aiming to is directly proportional to the damage that we can cause to it. So we really have a random kind of damage there on DNA. There is no preferential site. Um, but it also happens quite in a late moment of the inactivation, about 10 joules per square centimeter or about 20 joules per square centimeter. So that's quite late. Now, if we look at these graphs on the left side, I'll begin with the protein content one. We can see that as soon as we begin the inactivation process, the cell already begins to lose its proteins. This has been quantified by Bradford. We have this continuous trend of protein loss after PDT is being delivered to those cells. That's quite interesting. It has much more sense to what we're looking at the inactivation kinetics. When we look at the formation of carbonyls in proteins, which are the main product of protein oxidation, and to have some very early kind of response to APDT, the oxidative attack of APDT here. So we have a continuous formation of carbonylated proteins when we are performing PDT to those bacteria, which is very interesting. It's much closer to what we see in the inactivation kinetics of those cells. And also, down here on the left side, on the graph C here, we also measure the enzymatic activity of some proteins, especially the ß-lactamases, with the same method that we used for the last paper that I presented to you. And this is even more sensitive technique because if we have any kind of minor oxidations into those proteins, they might become inactive enzymes. So again, we saw the same kind of trend of enzyme inhibition in a dose-dependent manner that is quite close and aligned with the inactivation kinetics of those cells. So yeah, proteins seem to be the major player in the inactivation of cells through APDT. The last graph here is the membrane permeability test. We did it with N-phenylnaphthylamine, which is a classic probe to measure the permeability of microbes, of bacteria especially gram-negative bacteria. And what we see here is that when we expose those cells to APDT, it rapidly gets more permeable. The membranes get more permeable, but it saturates. And it has no kind of trend or correlation with the inactivation kinetics. And we saw on those last images, on the last slide, that the membrane never ruptures. So it wouldn't make much sense to be the inactivation being governed by membrane damage alone. And another interesting thing in this graph here is that it gets to intermediary kind of permeability. Here we have a negative control, which is just those cells in PBS. And we have a positive control, which are those cells exposed to 70% isopropyl alcohol for 15 minutes. The APDT exposed cells get significant differences from the positive and the negative controls. So it really gets to an intermediary level of permeability, which is not as bad as alcohol exposure, and is not just as sound as a non-exposed cell. So that's quite interesting. And if we look actually to some rationals here that we already have, which are, for example, what is the dry mass of components inside a cell? And here we can see for E. coli, Saccharomyces cerevisiae, which is a yeast, and mammalian cells, and we can see that for all those cells, around 50% of their dry mass is proteins. If we see these images here in the middle, those are another bacterium cells exposed to APDT, but this time with dimethylmethylene blue. And we can see that we cause all kinds of destruction in the cell. But we form those bubbles in it, which are commonly referred as inclusion bodies. They are just aggregated proteins. If we look at this table here on the right side, we can see that the highest, these numbers here are the the reaction kinetics constant. Okay, and we can see here that the highest constants are always for proteins, either when we are talking about hydroxyl radicals or singlet oxygen. And they're the most abundant components in a cell. So the fastest reaction that we might have in a cell is indeed proteins. So it's really reasonable to say that protein might be the primary target of APDT in a bacterial cell. Recently, I mean about 10 to 15 years ago, it has been discovered as well that gamma radiation, ionizing radiation, also kills bacteria by protein damage instead of DNA damage, as it was thought before these studies. So we might have pretty much the same kind of situation here for APDT. So proteins seem to be the most sensitive and abundant target for APDT. So we should be targeting at them. And if we want to go to some combination therapy with with some chemotherapy and antibiotic and so on, we might be rationally be proposing to target other structures. So, for example, we should be combining an antibiotic that acts on membranes or cell walls, because PDT will be focused into proteins. So we have a guaranteed synergy in between those techniques. So those are the main papers that I had to present. I'm just going to tell a little bit about other stuff that we have been doing for this time. So other than those 4 papers, we also published 17 other papers during these 5 years of my PhD. There are some quite interesting ones that are being pretty well cited. Many of them have Mike Hambling and Mark Wainwright as co-authors as well. I also participated to some book chapters. This one is from Sylvia, Marta's and Aguinaldo's book, the 2nd edition, actually. We are also present in the 1st edition. We talked about dosimetry on PDT. There's also the veterinary PDT book that I mentioned to you earlier. This one, Mike and I participated, I think, in 6 chapters. I participated to some others with Fabio and Marta and Chris. So we had we had 9 chapters in that book as well. I made some patents too. This one is a quite interesting one, which is a simple method to visualize acne, microcomedones especially, using fluorescence of protoporphyrin 9 and chitin that is present either in the fungal cells or the demodex. They are present inside those pores. So it's quite interesting method to see what kind of pores are infected by bacteria and which are just with demodex. We also developed a intraoral device to treat oral mucositis, which is one of the nicest applications of photobiomodulation. It can also be used now as a entire mouth decontamination system. For example, when we're going to intubate a COVID-19 patient, we can disinfect the person's mouth before getting the tube inside. We also have a system for disinfection of liquids using visible light, especially blue light, but we can also use that with UV light. It's quite interesting. We already have some prototypes of that being tested at Colgate-Palmolive. This is one that already became a product, but it's also out of the market because the Brazilian FDA doesn't like it, which is a system to disinfect N95 masks using ultraviolet light. What we actually patented is the way that we expose those masks, so they get suspended on little motors that keeps spinning them in front of one very high power UV light, so we can get all the angles in those masks just using one lamp. So it's quite interesting that. Got some awards too from the International Photodynamic Association that happened in Boston. From the I even got some money from Her Majesty the Queen going to the Leaders in Innovation Fellowship at the Royal Academy of Engineering in London. What was a very nice course that we did on scientific entrepreneurship. We got a gold medal for the University of São Paulo team in the International Genetic Engineer Machine Competition, the IGEM, that happens in Boston as well. We developed a bacteria that has some gene promoters that are sensitive to light, so they express some genes only when they're receiving some light. In that case, was blue light at 450. And I also got an award from the American Society for Microbiology in a congress that happened in New Orleans a few years ago. And I also opened my startup company, BioLambda. We began with equipments for scientific experiments to facilitate our cell exposure experiments. Within a few years, we already sold equipments to all 5 continents in the world in very well-renowned institutions and universities. So now we feel that our science is being in a much broader scale now, because I'm helping all these people to perform their own sciences. So it's very interesting. We also developed some anti-COVID systems for masks, decontamination, air, surface, and in rooms. Right now, we also with a project to try to help some hospitals here in Brazil. So we'll be donating a series of equipment for air purification to be installed in public hospitals here in Brazil. We already donated about 20 equipments, but now we need some partners and sponsors to help us scale this up. And so this is an invitation also to whoever can help us to face these complicated times that we're facing here in Brazil, please let me know. And thank you very much. So this is all I had to present. Hello? All right, so who should begin?
Nilsson, you're
You're still on mute. Let me just refill my coffee. Newton. Newton.
Newton, você tá mudo, Newton.
Sorry.
Aí.
Okay. Okay, então, please, the next, I invite the members to start the evaluation. And remember, Caetano, I cannot evaluate you. The first evaluator will be Professor Hamlin. Professor Hamlin, thank you for your participation. You have 50 minutes for questions, and this conversation could be informal, following question and answer, or by presenting all questions, whatever. Professor, feel free to make your evaluation, and thank you again for your participation.
Okay, so you want me to go first? Is that correct?
Sorry, Mike?
You want me to go first?
Yeah, you begin.
Okay, all right.
Punch me.
Well, congratulations, Caetano. You've done a phenomenal amount of work, and you've presented it, very cogently and, economically. You didn't sort of wander off to the point too much, so you got through it all. So well done on that. So I think my main point I want to raise with the whole set of experiments concerns the photosensitizer, choice of photosensitizer, the choice of the concentration of photosensitizer, and the choice of not centrifuging the microbial cells. In other words, leaving the vast majority of the photosensitizer in solution. Because as you well know, methylene blue is very prone to dimerize and oligomerize to form even more than two molecules stuck together, but the dimers for sure are a big deal. And several folks have shown that the lower concentrations of methylene blue work better than high concentrations. That's been shown many times. In fact, one paper showed that the optimum concentration of methylene blue was 3 micromolar, which seems astonishingly low. We've found it's probably somewhere between 5 and 10 micromolar. But folks that have tried 100 micromolar have been very disappointed.
Is this what you're talking about? Can you see my screen?
Yeah.
So there you go. We actually did that, but as a pilot study to establish what is the optimum concentration of photosensitizer for our system.
Yeah, so
we got something between 125 and 62.
Yeah, you find a biphasic dose response with a, you've got about 30 micromolar, which it could be, you know, who knows, it varies a bit, but so why didn't you use 30 then?
Yeah, so the choice there was really based on this graph here that I made. So we did some wide variations of photosensitizer concentrations. We can see that if we get it too concentrated, we might be suffering some filter effect. So cells in the bottom of the well that we are radiating are not receiving as much light as those on the top of the well. We can even, I even have a modeling of the exponential attenuation of light within these kinds of solutions in those concentrations. And if we get too little photosensitizer, we don't have molecules enough to excite to have a good reaction rate going on our solution. So we have this optimum point here, which is in between 125 and 62 micromolar. So I just got something in between here, which is 100. That was the choice.
Okay, so yeah, we found 100 was way too high, but leaving that aside,
that might depend on the volume or the optical path of your radiation solution.
Under these conditions, without any centrifugation, the vast majority of the photosensitizer molecules are going to be in solution. So when you activate them with light, you produce singlet oxygen outside the cells, which other people have done, you know, by attaching the photosensitizer onto particles and surfaces. So it's known that producing singlet oxygen outside the cell will kill the cells. But it's obviously less efficient than producing the singlet oxygen inside the cells. It's not really known if producing type 1 species outside the cells is effective. It may be, but who knows, because hydroxyl radicals don't tend to travel very far.
Yeah, but if we have, I'm sorry, Mike, if we have some superoxide or hydrogen peroxide, they can migrate for long distances.
Well, hydrogen peroxide can diffuse into cells very easily, but what concentration are you producing? I mean, you could have actually measured hydrogen peroxide concentration in your solution. It's fairly easy to do.
How is it? How do you do it?
Well, I mean, there are assays.
That's really interesting. I didn't know that.
Hydrogen peroxide is one of the most measured ROS there is.
But I mean, by using some DCFH or something like that?
I think it's a enzyme coupled reaction, I believe.
With catalase or something.
Yeah. Okay. It could even, you know, because anyway, I'm getting off the point. So, and I mean, your microscopy images were very nice, showing that the photosensitizer inside the cell is doing something. But the vast majority of your photosensitizer is not inside the cell.
So I'm not really sure after those images, because, for example, well, let me share this slide again, because we did not wash off the photosensitizer for those images, exactly to have it on the background so we can use it as a as a reference of intensity and lifetime. So you can see here that we have very good contrast in between what is inside the cell and the outside.
Because the photosensitizer in the aqueous solution is not fluorescent, because it's dimerized and aggregated. So you're seeing the fluorescence from the disaggregated photosensitizer inside the cell.
Not sure. Let me get some. Oops. This is absorber photons. Yeah, keep on. Oh, so here, Mike, can you see my screen?
Yeah.
Okay, so when we are here at 125 micromolar, for example, we still have some absorption band from monomers here at 660. It's decent one. I mean,
you're going to go right down to the 10 micromolar range before the monomers is the majority.
Actually, this data here shows that usually when we work with PBS, they get to equalized bands, let's say, at around 100 micromolar for the tour measurements, which is pretty much here. You can see that at 62, it already gets a predominant monomer band here.
And a lot of people have shown that if you put, some kind of detergent in that disaggregates methylene blue, the effectiveness goes way up.
Yeah, some SDS, even urea. See what I found with urea.
Many different things people have tried, and they all seem to work fairly well. So why on earth didn't you centrifuge them? There seems no reason to me why you would not centrifuge them.
Well, in my perspective, Mike, for example, when we're going to treat a wound, any infection with PDT, we just spray the photosensitizer there and tend not to wash it off.
You're doing a sophisticated mechanistic study.
Okay, in that point, I understand you. But what we do, we always try to mimic this kind of clinical situation. I try to do that, but it will be a very interesting thing to wash it off for the mechanistic ones. And even compare both situations, maybe. Without washing and with washing, how that might change the mechanisms. That should be a very interesting thing.
Because in a lot of our papers we did wash and no wash, and it's very different, actually, very different.
That's very interesting. Very interesting.
Because as you know, methylene blue is pretty poor photosensitizer. So any decent photosensitizer can kill logs of both gram-positive and gram-negative bacteria at less than 1 micromolar. A few hundred nanomolar, or even down to sort of 10, 20, 50 nanomolar for some of the real super ones.
Yeah. I actually chose methylene blue because it's the most widely studied and used in clinics.
I appreciate that. It's the one everybody uses. It's generally regarded as safe, and every pharmacy in the world stocks it. So it's
that was my motivation. That's a lot of work on those mechanistic studies to have like several photosensitizers being tested. That's a dream. That's very nice. I would love to do that, but we had to keep with our limitations and focus to one bacterium species and one photosensitizer.
No, I mean, I think that's it's fair enough to use methylene blue, but the question then arises is how widely applicable are the conclusions?
That's the point I wanted to get. So would we get those same results if we change the photosensitizer? If we just change the efficiency of the photosensitizer? So location is a very important thing as well, where that photosensitizer might be accumulated preferentially and how that could change the mechanisms of inactivation. Maybe if we get some photosensitizer that binds better to DNA, we might get some preferential inactivation through there. Let's say some sorolane, some acridine.
I mean, so one thing you didn't really consider at all mechanistically was lipids. So, I mean, there's actually a very good assay for lipid peroxidation, which is the famous T-bars assay.
T-bars, yeah.
Which has been done by dozens of people. And for a photosensitizer like methylene blue, maybe it's not particularly likely to go for lipids, but some of the the more lipophilic but still cationic photosensitizers, some of the engineered porphyrins or bacteriochlorins or these sort of things that are active at nanomolar concentrations.
No, we can just go, for example, to dimethyl methylene blue and get some decent difference.
It's
we actually did
several fold better than methylene blue for sure.
But it's also more toxic in the dark, right?
Well,
but yeah, we did some experiments with dimethyl methylene blue as well, and we took it to the transmission electron microscopy microscope to look at those cells, what happened. And like that image that I showed you with those graphs, those pizza graphs of the cellular contents. That was, well, let me get it. It's actually messier, but still it doesn't break the membrane. Oh, that way. Oh, here. So this is also Klebsiella pneumoniae, but on this E image here, it has been exposed to 100 joules per square centimeter using dimethyl methylene blue, and still we don't see any membrane rupture.
Well, I mean, I don't think we've ever seen a membrane rupture. What we have seen is big white holes in the middle, for sure. Yeah. Now, are these the same as you're showing there? Because they don't quite look like that. I mean, these are...
Yep.
I mean, you've got several little circles joined together. What we generally see is a big white hole taking up probably half the cell or something.
Yeah, this looks like smallpox on the cell, right? So they can either be empty bubbles or they can be aggregated proteins that are poorly stained by our, I think it was lead citrate that we use it to stain this thing. And they're poorly stained normally.
So is that known that aggregated proteins are not stained by lead for electron microscopy?
Yeah. A very classic case for that is, for example, when we use some biotechnology to produce large yields of proteins, of recombinant proteins in bacteria, in E. coli, for example. They tend to form some big aggregates inside their cells, and it looks pretty much like that.
I mean, what people have done is to measure biomolecules, which I think are mostly RNA that are released out of the cells. So, centrifuge the cells, and in solution, is RNA that comes out of the bacterial cells, which you can quantify.
Yeah, that's quite easy. You can even put it on a nanodrop spectrometer or something, and you can
I don't know what assay they use, but they definitely showed that RNA leaked out of the cells, and that is one evidence that the membrane has been permeabilized, I guess.
Yeah, shouldn't have RNAs outside. That's interesting too. We did some similar tests of those. I don't remember if we did that specific one. I can search through my data and take a note of that. But that's a very interesting, simple, straightforward technique to look at it.
Yeah, I think it's quite logical that proteins are going to be a major target. In fact, the proteins can be a major target of lipid peroxidation. So, if the lipid is oxidized first, it produces reactive species that then attach proteins.
Mm-hmm.
Now, whether they come up on your carbonyl protein assay, I'm not sure.
I don't think so, because this technique, we go through some purification after extraction. So we might be having mostly proteins there.
I mean, the malondialdehyde definitely attacks proteins, which you get from the lipid peroxidation.
So you should have like an extension of the chain reactions caused by radicals going from lipids to proteins.
Yeah, I mean the two really reactive products you get from lipid peroxidation are malondialdehyde and nonenal. And these are well known to be detectable conjugated to proteins.
Interesting. That's very interesting.
I'm not sure whether this is a big deal in microbial cells. All this is really being done in mammalian cells.
But I mean, if those microbial cells have the same initial lipids as the mammalian cells, which is quite often, depending on the lipid, they should have the same types of reactions. Yeah.
Somebody changed the lipid composition of microbial cells, I think. Depending on what you put in the medium, you can change the concentration of unsaturated lipids.
Oh, yeah, I mean, each species in each phase should change its lipidomics. Especially talking about microbes, they should have a very large variation.
Yeah. So if you can raise the concentration of unsaturated lipids, then they will be much more susceptible to lipid peroxidation.
Definitely. Yeah, making some T-bars was actually in the plans at the beginning. But we got some bad kits to test. We did not have much funds to buy other kits, and we kind of left it for our next time. But T-bars is quite a nice thing. But actually, I thought that, okay, we might even be forming some lipid peroxides. But what actually imports to me mostly is how is the function of those membranes? Are they still functional? Are they still working as membranes? Are they being permeabilized? Or they're being ruptured? What could be happening
in
functional terms?
An assay for membrane polarization, I believe. So the healthy cells have got a polarized membrane. And when the membrane is depolarized, you can measure it. I think it's a fluorescent dye, I believe.
That's rhodamine. Rhodamine 123, I think it is.
So I think you get membrane depolarization before you get permeabilization.
Mhm. Let me take it. You can keep on talking, Mike. Just taking notes here.
And also, another thing I think people have measured is respiratory chain activity. Because that is one of the things that goes quite early, I think.
What? I'm sorry, I missed that one.
Respiratory chain activity in the bacteria.
Respiratory. Yeah, that can be measured with the membrane permeability as very in an indirect kind of term. But that's what people usually do for mammalian cells. They use this rhodomain 1, 2, 3 and see the polarization of mitochondria. And there you can have this correlation on how the respiration is going. Otherwise, we could even maybe use methylene blue with some bugs and glucose.
Yeah, I mean, certainly, you know, with Candida, they've done that a lot, playing with the metabolism. Whether they've done it with bacteria, it certainly works well with Candida. You know, the folks at Rochester did that a lot.
Yeah, bacteria should be simpler. True. Oh.
But I think that panel of 4 graphs you showed was quite convincing with the, you know, the different shapes of photoinactivation. The only one that would have been nice to have on there was lipid peroxidation as well, just to see what shape that took.
Yeah, that's nice. I agree with you.
So why do you think Klebsiella is so resistant?
$1 million question.
Because I would have assumed it was similar to E. coli. I have to say. I mean, you know, microbially speaking, it's very similar.
Yeah.
I mean,
go ahead.
We know that Acinetobacter is highly susceptible. We've known that for a long time.
It's pretty much similar to E. coli.
Yeah, Pseudomonas is more resistant, but
Klebsiella is even more.
Way more resistant than Pseudomonas, which is quite interesting. No idea. What about
that's a big question that we have, and we had some hypotheses that we got to that. So first one, if we look back at those graphs, let me get it here. I'm sorry, not COVID. So if we go back there, here, we have a big difference in what is on the left side here on this scale 1 and what is on the right side here on this scale 5. In respect to to bacteria, if we look at what we have here, Abelmania, Ificalis, Efficient, E. coli, and Staph aureus. And on the other side, E. aerogenes, now it's K. aerogenes, but whatever. K. pneumoniae, Pseudomonas aeruginosa. Everybody here on the right side has a capsule. Nobody here on the left side has a capsule. That's one point.
Right. Okay. So some of these graphs are blue light, right?
Are what? Sorry?
Some of them are just blue light.
No, they're all MBPDT.
They're all MB, okay. Because when you did the blue light, you didn't measure intracellular porphyrins, which would have been the obvious thing to do, because they kind of go together.
And in that case here, look at this comparison here of ABL, here the graph below. Then we even have some difference in between strains, and that should be intrinsically linked to the content of this strain, the ability of this strain to accumulate porphyrins and whatever else it might be absorbing light. But when we use methylene blue, there is no such thing. They all behave the same, and we only have difference in relation to different species and not different strains.
So, I mean, you showed that blue light could inactivate beta-lactamase, but that is clearly ROS being generated from intracellular porphyrins.
Definitely.
I mean, if you took beta-lactamase as a soluble enzyme, blue light wouldn't do anything to it, presumably.
They kind of are a soluble enzyme.
Sorry?
So we can assume that the ROSs are migrating inside the cell. They're diffusing.
Yeah, absolutely. I mean, the beta-lactamase is on the outside, I guess.
So here in this specific situation, they stay in the periplasm. So let me get some, I don't know, they go ahead. I think this one is the best to show. So they stay in the periplasm, in between the outer membrane and the inner membrane. That's where they accumulate all those beta-lactamases in a gram-negative.
It's only gram-negatives that have beta-lactamases, is that correct?
No, gram-positives also have them, but they just secrete them. They throw it out.
Oh, okay.
Mm-hmm. So they have it accumulated in this region here. And for that test that we made on beta-lactamase inactivation, leave just Pinkman there. In this case here, we extracted all the beta-lactamase from those cells. Those tests, this quantification, I made out of those cells. Like what we do is we sonicate those cells. Here we use it Klebsiella as well. So we sonicate those cells, and then we extract the supernatant to make those measurements and leave all the cell debris outside of the reaction. So they're soluble, and they do not require ATP or anything to work. Okay.
Right, you could have extracted the beta-lactamases first. And then
yeah, I did. Yeah, to expose them to APDT without.
I mean, you could have done that.
Yeah, but I doubt it would work.
It would have been quite interesting. I mean, obviously, you don't have unlimited amounts of time and money, but it would have been quite interesting to do the, the proteins, the DNA, the membrane permeability, and the lipid peroxidation with blue light alone. That would have been quite interesting.
Yeah. So that's the project of my friend and sort of student, which is Carolina. She's at Tianhong right now. She's been there for over a year, and she's the one who is going to make the mechanisms of blue light and show them to Niels Finsen.
I mean, I'm guessing the results are going to be pretty similar. That's my guess.
Not really. And the crazy stuff is that changes a lot according to the species that we were talking about. When we looked at, for example, DNA degradation and membrane permeability as well, it changes a lot from Pseudomonas to E. coli to Staph aureus. Changes a lot, the pattern. Because we're depending much more on the physiology of the microorganism that we're working with.
Also, the porphyrins change a lot. That's the key.
Yeah. Some have more soluble porphyrins, some have less soluble.
Some just don't have any. I mean, Ephicalis, for instance, just doesn't have the enzyme, so it can't have any.
Interesting. Didn't know that.
Yeah, it needs blood in the medium. It gets its heme from blood. So it just doesn't have the biosynthetic enzymes.
Got it. So it's a kind of fastidious characteristic of it.
Yeah, there's another name, auxotroph. Is that the name? There's a special name for it anyway.
So another interesting thing here that separates those two major groups here, it's another theory that I have on the production of glutathiones. Those are very strong antioxidants that we have inside our cells. Some cells have more than millimolar concentration of that stuff in a reduced form inside the cell, in the cytoplasm. And that's active against even single oxygen and hydroxyl radicals as well. Okay? And if we look here, Staph aureus doesn't produce any. Enterococci are some of the few gram-positives that produce glutathiones, and they're quite tough gram-positives to APDT. Gram-negatives have some variation. Some produce more, some produce less. Abelmannii and E. coli seems to produce relatively less glutathiones, but those Klebsiella, Enterobacter, and Pseudomonas, they seem to produce quite a lot of those. Another interesting thing, Pseudomonas might be even using some of its pigments to scavenge some of the RSs.
Could well do. But some of these Pseudomonas pigments are actually photoactive.
Yeah, that's one of the reasons that they're so sensitive to blue light.
Yeah.
Because those pigments absorb lots of blue light. So that's a very interesting point. But when we are working with red light, it seems to don't have as much impact.
And of course, Staph aureus has a lot of these ROS
carotenoids,
staphylos and things.
Yeah, so the interesting thing about Staph aureus is that it has a lot of ROS quenchers in the membranes, not in the cytoplasm. That's a very interesting thing. Because all those carotenoids, those carotenoids are very lipophilic. They don't like water at all. So It has all of them concentrated on the membrane. And a very interesting slide. I made some transmission electron microscopy images of Staph aureus irradiated with PDT. And it's really interesting because it forms inside the cell some patterns that look like fingerprints, some like an onion inside the cell. And I was showing it on a IPA meeting in Coimbra, and Professor Melnik from Israel, that's a guy from the old times. I was showing those results to him, and I said, I have no idea what this thing being formed inside the Staph aureus is. And he said, oh, those are mesosomes. I said, oh, come on, you already knew that straight on. And yes, it's a sort of intracellular ROS defense for Staph aureus. So since it only have those very strong antioxidant carotenoids on the membranes, what it does is to form membranes inside the cell to protect its intracellular components. So that's what it does. It's pretty cool, I think.
All right, well, I think I've probably used up my time. I don't want to carry on this too long, so why don't I pass on to somebody else?
That was very nice. Thank you very much, Mike.
Well,
and if you have anything else, if you have anything else to add afterwards, we can talk and so
on. Are you listening?
Yes.
Okay. Professor Michael, thank you for your participation. Very interesting question to be considered to the next paper, the last paper of Caetano. Thank you so much. Now, Professor Mark, please, could you make your evaluation?
Yeah, I'm okay. Yeah.
Okay. Thank you so much for your participation.
You're very welcome.
Hello.
Dude.
You
really need to start putting in some work, man. There's hardly anything there.
It's been intense.
So one of these days you might get about 2 minutes sleep or something,
I hope so.
So that was as expected. It was a kind of superb presentation, and there's so much there. There's certainly more than one PhD there, that's for sure. Okay. So obviously I had a huge list of questions, and Mike has has asked about 3/4 of them. So,
come on, Mike, should get more for yourself.
I mean, I think that was always going to happen. So I have just kind of rearranged what I've got left. One of the things that I did wonder about was the abstract of the paper that we're looking at, the paper 4.
Okay.
There's a sort of a quote on there on line 10 of the abstract, if you want to bring that up and people can see it.
Let me get it just a second. So, nope. Thesis. Final. Thesis. There you go.
Yeah, it's on about page 48, I think. Oh, yeah, there we are. Okay, yeah. So on line 10, it says, where am I? I've lost it now. 1 second. It starts, yeah, towards several biomolecules. And then researchers simply assume that all cellular structures are equally damaged. We need to think about how we write that, because as a chemist, if you think about, say you take singlet oxygen, and you think about the different types of cellular structures. So you think about the, say, proteins, lipid, and DNA. I don't think it would be a natural assumption that all cellular structures would be equally damaged or damaged at the same rate. And I suspect that what you've found about the proteins being the most important target, or the critical target, it's probably due to the rate of reaction. So that I think we need to, within the abstract, if we're trying to get this published in something like Nature or Science or something like that, then I think we need to be as clever as possible here.
Yeah.
So we need to have a think about that. Just in very simple chemical terms, if you're reacting a standard reagent with 3 different chemical types, then it wouldn't be a natural assumption they'd all react at the same rate. Okay, yeah. And these are obviously quite different. Proteins are different to lipids, lipids are different to DNA, and so I think we need to think about that. And further down, I think it's line 14. You're talking about where are we? 11, 12, 13. That can't be right. Just give me a second.
Take your time, don't worry.
Yeah, sure. We talk about membrane permeabilization. Obviously, if we're talking about bacteria, then are we talking about gram-negatives or gram-positives? Because obviously gram-negatives are going to have, there are 2 potential membranes there that you could be talking about, whereas with gram-positive there's only the 1 membrane. So again, we need to be, do we need to expand that, or no? I don't know what the answer is, but I think that might be a little bit vague. So we need to kind of think about that. From that comment just now, if you think about the gram-negatives, where you've got 2 membranes, you've got your outer membrane and you've got the cytoplasmic membrane as well, we haven't really considered what's between those 2 things. So I mean, you've got peptidoglycan for a start off. You've got various kind of trans or connecting membrane bodies in there, porins and things like that. Did you think about anything to do with those sorts of structures, with peptidoglycan, for example?
That's a tough kind of analysis. I'm not sure how to measure peptidoglycan damage, cell wall damage in general, but that's a very good observation. If we look here in this photomicrograph of those exposed cells on the TEM analysis, you can see that the periplasm is being expanded, right? So what it really seems that is that the structures in between the inner and outer membranes are being damaged, so they're getting just floppy ends. I don't know, they're just losing up.
Yeah, yeah. I mean,
I
obviously,
can see that on the images. I don't know how to quantify that.
yeah. I just wonder if it needs to be mentioned, even if you can't quantify it and didn't, obviously didn't try and quantify it because we don't know how to do it. Obviously, peptide glycan has a considerable amount of protein in there, so we presumably aren't discounting the idea that you could damage that via a similar mechanism. So again, that would be an interesting, an interesting thing to talk about. Just 1 second.
All right.
Okay. Just needed some more tea.
Another interesting point is that here we presented this membrane permeability data based on n-phenylnaphthylamine, okay?
Yep.
Which interacts mostly with the inner part of the outer membrane, okay? We also made the same test with propydium iodide, which gets the signal from intercalating with DNA.
Yeah.
And we couldn't get any signal. So the thing is, I kept thinking about some stuff. And for example, one thing that could happen is that we're just damaging the outer membrane so that the n-phenylnaphthylamine can get inside and get fluorescent and bring us the signal that we're looking for. But the propydium iodide cannot get into the inner membrane and interact with DNA, which is inside. That's one thought, maybe, I don't know. But another problem that we have with propydium iodide is that it can undergo through some fret interactions with methylene blue. So, because this thing emits red light around the same absorption at the same band as the absorption band of methylene blue. So we could be quenching its fluorescence, and both of them should be intercalating with DNA. So we have an optimum distance for this fret phenomena.
Yeah.
So we thought a lot. We couldn't figure it out, separate those two influences. We just decided to keep outside this propydium iodide data and stick with this NPN data.
Yeah, I mean, it kind of goes across what you said to Mike about perhaps using acridines to look at DNA damage, because then you cut out the requirement for methylene blue then, and then you could still get the fluorescence.
But acridines will emit close to 600 nanometers yet. We'll get the dimers band, but won't get this close interaction, right, in the DNA.
Yeah.
That's a nice one to try.
Yeah. It's something you might want to look at. Okay. That's fine. So you asked about the blue light, so that's gone. Capsule you did mention. What do you think the capsule adds in terms of bacterial defense?
I have no idea, because I mean, dude, capsules, they're highly permeable, right? And they're poorly stained normally. Even though they have some strong negative charges, it could sequester some of the methylene blue molecules. But I don't understand how they can influence this much. And look, another thing that I omitted to Mike. So here, down below, we have Cryptococcus neoformans and Candida albicans. Candida albicans is much more tolerant than Cryptococcus. And Cryptococcus is the one who has the capsule. Candida doesn't.
Yeah, that doesn't help, does it, at all?
Not at all. Even
Yeah.
though Renato, I think when he was at Mike 10 years ago or so, he did some experiments with a strain of Cryptococcus neoformans that had a deleted gene for capsule. And he
Yeah.
saw there that it was more sensitive to a PDT than the wild strain with the capsule. So still, it's a kind of crazy assumption. I have no good idea on why the capsule should be bringing us problems. That's why I'm not saying that it is the capsule, but we have this crazy coincidence on the bacteria. Another quite nice one to test, in my opinion, that I could never get my hands into some good strains, is to make the same test with Streptococcus pneumoniae. So we can get the same species with and without the capsule and compare them directly and see if this thing really does some difference.
What about the structure, if you like, the chemical structure of the capsule? It's mainly polysaccharide. Alginate. Okay. So I guess there are different forms of alginate in some way. It will be chemically similar, but there will be, I guess, it will be different molecular weights, or somehow there might be different structures of capsules. They won't just be the same structure on every bacterium, and obviously different bacteria will be different.
And yeast.
Yeah, again, so I don't think it's, I think you could expect some variation.
Yeah, me
so
too, but
it doesn't make it easy.
If you have some insights on how, we discussed that before.
We did, yeah.
But I didn't have some mechanistic insights on how that thing should be working, because it's permeable.
Yeah, that's right. So picking up on something that I was going to ask, that Mike asked, I just wondered if we'd looked at the whole thing. You talked about not spinning down the cells. So you didn't say you've got the, because that's exactly what I would do also, because I would think you want to mimic as far as possible the clinical aspect. So you apply your material. So you could, of course, say that in some cases, I've seen some papers where the photosensitizer has been applied, it's been left there for a certain amount of time, and then the wound site has been rinsed in some way. So quite a lot of dental applications do that.
Yeah, especially for dental, because of the statics of having stained blue teeth, right?
In this day and age, everything goes, man. Don't worry. So, what is the big disadvantage to leaving that methylene blue kind of around the cell? What do you think is the big disadvantage to that? Or do you think it's a disadvantage?
Only in mechanistic terms. For real life clinical applications, I think we should leave it outside, because it won't make much change in relation to the filter effect of light penetrating into the infected tissue. And another very important thing that everyone who has put their hands to work on PDT has probably seen is that as soon as you begin giving some dose to that solution, the cells begin to uptake more and more and more photosensitizer. So for example, if we get this Klebsiella cells here and expose them to 5, 10, 15, 20, and 25 joules per square centimeter, put all of them inside Eppendorf, centrifuge all of them, and look at the pellets, we would have like a degradé of colors there, being 0, the most whitish pellet, and 25 would be like a dark black pellet with tons of methylene blue inside. So if we take it to a clinical situation where we are radiating the wound and we want to get the most effective disinfection as possible, we should be leaving the photosensitizer there, because those cells still are going to uptake more and potentially amplify the inactivation effects of PDT.
Yeah, I completely agree with that. I think, from a chemistry point of view, you have a dynamic situation. So you've got your solution outside the cell. Some of the molecules will leave and go into the cell, but you still have a reservoir there, so that if more molecules can go in and obviously will cause more damage, then that's another good reason for leaving some methylene blue outside there. The aggregates argument, again, aggregates are still dynamic, and they can be thought of as kind of slow release of methylene blue into solution, which then can get into
like a buffer.
So, yeah, precisely. So, I think you're quite right to do it in that way. I think it is also interesting to look at wash and not wash.
Yeah, especially for the mechanisms. That should bring some further insights, definitely. But for the real world situation, I would not recommend to wash it off.
No. I mean, that's.
I wouldn't even recommend to leave a pre-irradiation time for the wound. I would just put the dye and start irradiating straight away.
Okay. I mean, obviously, the more damage you cause to the cell, the more likelihood there is you'll get more methylene blue taken up, or
everything gets faster. Yeah.
Oh, that's good. Okay, that's one more. And yeah, again, thinking about the proposed paper 4, talking about an adjuvant effect or potential adjuvant effect of photodynamic plus a conventional antibacterial or an antifungal if it was a fungal infection. How do you think that would work clinically?
So clinically, the guy would already be under antibiotics. We are not recommending antibiotics at any kind of protocol, right? Just keep them on antibiotics and add the PDT. So let's suppose that the guy has a wound that is infected, diabetic foot wound. He's taking some antibiotics by default, and then we do some PDT. In that wound, there might be some drug-resistant bugs in that are resistant to the antibiotics that this guy is taking. But when we do the APDT together with the antibiotics, we might have a chance to have this antibiotic working on those cells again. And we already did some experiments and published some papers from the time when I was at Hamblin, and showing that we can just give a really sublethal dose of APDT, like killing 0 bugs, 0 logs, and then expose those cells to antibiotics, and antibiotics start to work. You can even think of a crazy situation as this, as to have a Klebsiella with KPC or whatever, and give them some mild dose of PDT, and then give the person will be under some antibiotics, let's say penicillin, or something like that, amoxicillin. And this amoxicillin would work with a Klebsiella pneumoniae with KPC. That would be nuts, right?
Yeah. Oh, yeah. I mean,
we would revive some antibiotics, give them another shot.
Yeah, I mean, I guess it's also possible to apply antibiotics locally. Rather
as well. Combining the formulation of the photosensitizer.
Yeah. Is there anything that you could do in the lab to kind of make sure that the two, say a ß-lactam and methylene blue, to make sure that they would work together, that there wasn't some kind of contraindication? Or
we can do a few tests. We can do, I think Carolina has been doing some of those there at the Wellman with Tianhong. That can be done in vitro through MIC tests. right? No problem. But we already published it quite a long time ago with Chibebe, an in vivo model of this combined therapy.
Mm-hmm.
And we did that with Galera melonella. I've even tried to find it here. There you go. We did that with VREs and also with Candida. And look at this cool thing here. Oh, here it is. Yeah. No. Here it is. Yeah. That's the graph.
Yeah.
Oh, we're using some VRE and try to treat them with vancomycin. Doesn't work, right?
Mm-hmm. Mm-hmm.
So, PDT alone, we did a very low dose. Didn't work at all. A little extension in the survival rate of those infected Galera melonella. Vancomycin alone, nothing to. And then when we do PDT plus vancomycin, we have a relevant extension of survival rates of those Galera melonella. Yeah.
Beautiful. Beautiful.
So we have some synergy there, and we are treating VRE with vancomycin. That's pretty cool. Of course, we can optimize parameters and so on, but it does work.
Yeah, yeah, for sure. Okay, no, that's good. I guess the only thing you would perhaps need to look at is are the antibiotics that you choose stable to singlet oxygen or oxygen radicals, or because that's
definitely. Yeah, I have one antibiotic in mind which is very resistant to oxidation. And I wanted to make some formulations with that. For example, if we use some phenols, chloramphenicol, fluorphenicol, they're really stable stuff. You can even autoclave them and they'll be just fine. And they should be inhibiting the production of new proteins, since we are degrading all the proteins with PDT, and we can prevent the microorganism to produce new proteins. That should be a quite nice way to get them, right?
Yeah, I like that.
Yeah,
right.
So here so here you can see the I have all the formula written down. So here you can see the same phenomena that we saw with those VREs, but with Candida. A Candida that is resistant to fluconazole. There's a very different resistance mechanism that is mostly focusing into efflux pumps. And we show the same thing. Fluconazole alone, PDT alone don't do much. But when we combine fluconazole with PDT, that's the gold. And it doesn't matter the order that you do it. It can be fluconazole first, PDT later. It can be PDT first, fluconazole later.
Weird.
Works just as well.
Brilliant stuff. Very good. Very good.
This is old, dude. It's from 2013. That's pretty cool. I really love those.
Still good. Still good. That's that one done. And so that one's done. So the only one that I've left is I've got two actually. So you talked about and I remember you talking to me about this, I don't know, two times before in San Paulo, quite a while ago anyway. You talked about the results you got with DNA and how it was chopped up into tiny pieces, which is impressive stuff. And I haven't actually seen anything in the literature on that. Such a good thing.
I was repurposing some techniques. They generally not use it for that. Yeah.
Well, is that likely to give you any regulatory problems if you're trying to get materials through?
Yeah, I agree. I should have some resistance there. Okay, you're killing an infection on me, but causing a mutation that will give me cancer and so on, and blah.
Yeah.
But I mean, yeah, we do. But the main target that we're going is proteins. And if a cell doesn't have good proteins and with some broken DNA, it will die. It will go through apoptosis.
Yeah.
Okay, much before it can generate some cancer. But if you say, okay, but if I have a cell, there's a particular thing that just received a minor amount of photosensitizer and that generate a very little amount of ROS, it just damaged a little portion of DNA that could cause some oncogenic mutation. Yeah, and there is a chance. But good thing about PDT is that it's a kind of new thing because it's not in the mainstream medicine, but we have been studying this thing for 100 years, more than that, 120 years.
Yeah.
Do you ever had any experience of tumor formation after antimicrobial PDT? I don't think so.
No.
Never, ever, ever. But there is an interesting story there. There are some papers from Melnick in the 70s when he was treating herpes with phenol red.
Yeah,
Remember
it
those?
was a bit lower down than there. It was he was treating genital herpes, I think, rather than normal herpes. So.
Oh my god. Yeah, there is a crazy story there. I even talk there. You can find this story on the internet, but I actually talked with Professor Melnick live about it. He confirmed it that he had a postdoc at the time that was doing those experiments, and he raised up that very same question. And he did that same thing in some important conference. I don't know if it was some ASM conference or something. And at that time, APDT was kind of getting trendy to treat this kind of infection, but this declaration that he did put everything back until you began working with those drug-resistant bugs in the 80s, early 90s. So that was a major push back in the 70s. Maybe the thing didn't took off because of that.
Yeah, I mean, a lot of the problem with that, I suspect, was that they were using things like UV activation.
Yeah, that's complicated.
So I don't think that they had done the kind of UV on its own controls, or so if you use UV and neutral red or UV and proflavine, then you may get problems. Yeah. So so you're right. It was looking good, and then suddenly it didn't look so good at all. So yeah, I mean, whenever we get close to clinical use of these things, the DNA interaction thing always comes up. So you in your future work, which obviously is going to be a lot longer than my future work,
you're much healthier than I am.
I wasn't talking about health. I was talking about longevity. So you would need to have a good response ready. I mean, it begs sense to think about it and try and work it out, and or even just don't mention the DNA until it's too late. I don't know.
Yeah.
My only other question was, do you think there's any application of this in terms of materials rather than a direct therapeutic approach?
You mean to have some like plastic that is bound to some photosensitizer and can work as that microbial surface?
That kind of thing, or at least a particular.
There are people doing it already. I just don't really see. I mean, now I'm Caetano speaking from the company side.
Sure.
I'm not really sure about the market for that kind of thing, since right now, for example, we have some silver nanoparticle doped materials, and they don't even need light, and they will do the same effect. So even though it might work, I don't know if there is a market for that.
Okay, no, that's fine.
Yeah, pretty much like how Novartis killed Visudyne with the anti-VEGF stuff.
Sure. Yeah. It makes
sense.
Have
a simpler thing that is just as cost effective, so let's use the simpler.
Yeah. So until we see significant silver resistance, there's no. Okay. So as I said, with my questions coinciding rather a lot with what Michael had already written and asked, I'm out. That's the end of my question. Thanks for that. That was all very good.
Thank you, dude. You're awesome.
You're welcome.
Okay, Mark, thank you for your consideration. We have, interestingly, we have the capsule structure of K pneumonia and K aerogenes, but we could talk about this later, but it's a very important point. Okay. Now, Professor Jorge Sampaio, thank you for your participation. Please, you could start your evaluation.
Okay. First of all, thank you very much, Caetano and Newton, for choosing me to see. It's very good to see this excellent work. It's very easy to talk about it.
Thank you, Professor.
So, I'd like just to ask some questions. The first point is at the title page, it should be postgraduate, not graduate program.
Okay, sorry. Going there.
At the title page, this is postgraduate program. So,
all right. Got it.
And I would like to talk with you about the draft article, the article 4. And the title is protein oxidation. Do you really think that the experiments you performed, you can clearly show that there was protein oxidation?
You mean about what kind of damage to protein, or if it's indeed the protein that is causing that is the primary cause of cell inactivation?
No, because the title of the draft manuscript is called protein oxidation. You are.
Yeah. So
you are saying that.
Yeah, antimicrobial photodynamic therapy. The photochemical mechanism of action is by the production of reactive oxygen species. It is going to be oxidation. Okay. The real question was is the protein, the membrane, or DNA or RNA oxidation that first leads the cell to its death. And according to the data that we have on comparing all those kinetics, all those response views of damages that we are causing to each one of those structures, the one that best correlates with cell inactivation is protein. And also based on the chemical arguments on the reaction rates and concentration of each component in the cell, the reaction velocity should be aiming towards the proteins. So it really remains as a major point to look at when we are talking of cellular inactivation. I do believe that with our data set, we can state that protein oxidation is the primary cause of cell death. That doesn't mean that we won't have other kinds of damages and oxidations within a cell, but what actually is the first one to take a living cell to die is the lack of viable proteins.
I point that because none of your experiments, you showed that there was protein alterations, but you didn't show that they were oxidized.
I did show.
This is the principle of the method, no?
Yeah. No, protein carbonylation is the main product of protein oxidation. So, and that's one of the major graphs there in this study. Let me share my screen again. Just give me a second. Here, on these graphs here, it's the one with the letter B, carbonylated protein. This is a product of protein oxidation. It's the most classic one. It's what it's used as a gold standard.
Okay. So another point is about the target used for PCR. For PCR, you tested targets that are located on plasmids, and for the FGE, we only saw the chromosome. Perhaps you should test also a target on the chromosome, no? Or talking about PCR.
Yeah, that's an interesting point. However, it doesn't really matter in my thinking whether the genes are in the plasmids or in the chromosome, because they will be in the same compartment. They'll be just as exposed to the photodynamic reactions as each other. So there's not really a preference of target in between plasmid and chromosome, both of them. And according to this data here, where we saw that a target that is twice as big as the other is twice as sensitive as the other as well, we can see that this is a very good indication that it's a random process, that all DNA is receiving damage. And according to the size of the target that we will have more hit points in that target than the other.
I think in order to say that, you should have used 2 pairs of primers. One pair that has a 800 product, for example, for KPC, and another pair that has a 400 pairs product also for KPC. They were located on the same plasmid.
For this very same gene. Yeah.
Because the problem is, were they located on the same plasmid? You have thousands of plasmid copies on a single cell, and depends on the plasmid. And depending on the plasmid, you may have this difference. Another point I would like to ask you: did you test anaerobes? Did you test bacteria in biofilms?
We have worked with bacteria in biofilms before. It's a very trendy kind of study for antimicrobial PDT. It does work really well. Of course, we lose some efficiencies, not as good as for planktonic suspensions, but it does work, and works both by inactivating those cells. And another interesting point of working on biofilms is that we begin to detach that biofilm from a surface. So for example, I have a biofilm on some surface that is grown there. I expose a few samples to different doses of PDT, and as you go to higher and higher doses and put those samples in a scanning electron microscopy, for example, for visualization, you begin to see that the biofilm gets smaller and smaller and smaller, and at the very high dose, you'll get just a flat, clean surface. You'll detach everything from the surface. 2 points might be happening there. We might be solubilizing the EPS, the extracellular polysaccharide matrix, and the proteins that actually attach those cells to the surface that is colonizing. So those things should be happening both at the same time. So we are killing those cells by the same mechanisms that I showed here in suspension, but at the same time, we're cleaning off, literally cleaning off the surface that was colonized by that biofilm. However, in this study, in my PhD study in specific, we did not focus on biofilms. We were focused to these other points in the kinetics analysis and some of the biochemical mechanisms. So we needed some cleaner, more simple situation to work with to eliminate some variables, because biofilms, are quite tough to work with. They have some good variations on the growth techniques and so on. But yeah, we have studied that with Candida, Pseudomonas, Cinetobacter baumannii, and even Renato, who might be watching us here, tested that with anaerobic. Is it anaerobic? No, it's microaerophily bacteria, which is Actinomyces concomitans, Agregatibacter Actinomyces concomitans, right? The AA, that very tough severe periodontitis causing bacteria. Working with anaerobic bacteria, it's kind of complicated because of the environment in which this bacteria is. That environment naturally won't have much oxygen, otherwise they wouldn't be thriving there, right? And we do need oxygen to run our photodynamic reactions. So if you work with them in some planktonic suspension, some in vitro experiments, you might have some good results. But if you don't have some pre-treatment technique to work with some biofilms of anaerobic bacteria, I really doubt that you could have some very good results with that. Some techniques, for example, people are treating chronic periodontitis with PDT, okay? There are lots of anaerobic stuff in there. Tenerella forsythia, Treponema denticola, Porphyromonas gingivalis, they're all anaerobic. So what can you do to actually prime that community there in the periodontal pocket of a person? Before doing the PDT or even adding the photosensitizer, you can just put some hydrogen peroxide in there. So you oxygenate that environment and allow the photodynamics to run afterwards. We can even mix both the photosensitizer with hydrogen peroxide and irradiate the whole thing with a oxygenating solution. So you need to run some strategies there. Yeah, it's possible.
I asked about anaerobes because in diabetic lesions, it's very common to have mixed microbial flora.
Yeah.
So
it's just like an environment there,
yeah.
A whole ecosystem, right?
So it may be a complicator in the clinical trials.
Yeah. Professor Tardivo, who is our partner physician, is an amazing person, has been doing this photodynamic treatment for diabetic feet for, 30 years by now. And he has like a success rate of 95% of preventing amputations. He actually does the priming technique in some of the cases, especially where it has to irradiate some perforating disease or something that has some collabated blood vessels. So what he does is to inject some methylene blue with hydrogen peroxide, and it helps in two situations. One, to oxygenate that environment, and also if you have a collabated vessel, it expands the vessel and allows you to go inside with a fiber or something to irradiate that cavity properly.
Yes. The last point I'd like to ask you, you say you showed about protein loss.
Mm-hmm.
Why you evaluate the protein loss just looking at the bacteria? Why did you look at the supernatant?
I think we had that control as well, but it was not as concentrated as the pellet that we could recover after the experiment. So our data was really noisy, messy, wasn't really good because of the concentration of the protein. We really need a lot of protein to do those quantifications. So working with the pellet was much more precise and reproducible, and we stick with that. But the main source of protein loss is that these techniques of quantifying proteins, we're mostly looking at the peptidic bond, right? And when we begin to depolymerize those amino acids and also oxidate each one of those and so on, we begin to lose the chemical means of making those biochemical reactions that we use to quantify proteins in a medium.
Yeah. So that's all I would like to ask you. Thank you.
Thank you very much, Professor.
Congratulations.
Thank you. Newton?
Okay. Okay, well, thank you for all members for this important contributions. Summarizing the main points, we agree with all considerations. It's very clear that lipids are critical target, and in the future, similar investigation on the mechanism could be performing on mycobacterium cells, for example. Regarding difference on PDT activity on gram-negative cells, I also believe that there is a tolerance by K pneumonia and K aerogenes, most likely mediated by the capsule.
Yeah, that's a very crazy thing that I really would like to investigate. Yeah. What causes that? What is the cause of capsule interfering with that? It's really crazy. Yeah.
Caetano, Fernanda have the chemical structure of all capsules of K pneumonia. After sequencing, we obtain the structure. It's a very interesting result, and we could use for your paper.
Nice. I really want to see that.
Remembering that we have hypermucoviscous K pneumonia, K1, Caetano, hypervirulent K pneumonia that produce a lot of capsules.
Yeah, that's another thing. We tested those. Yeah, we tested those. We published a paper on that, and we could not find any difference in between those regular K pneumonia and those hyperviscous mucose. That's complicated. Hypervirulent K pneumonia. So we could not find that difference there. So might be something inside the cell instead of just the capsule, and that's a coincidence of the capsule. I don't know. I'm really confused on that.
On the other hand, the efflux pump could be a resistant mechanism to sensitizer, methylene blue, I don't know. Indeed, quartz pumps can confer resistance to ethidium bromide, acridine.
Yeah, I also had that same hypothesis in my mind. But I mean, when we did this study on the MDR and XDR strains, and we had lots of resistance mechanisms that were mediated by efflux pumps, and we did not have any difference in between different strains of the same species. So that kills off that hypothesis in my mind as well. So still, is it some efflux pumps that is just present in this species? Doesn't matter of the drug resistance profile? I don't know.
Okay, Caetano, we have the information for gram-negative bacteria. We have the genome and deduction of efflux pump. We could see. We could see this.
Yeah.
Okay.
Renato again. He did some experiments in the past with verapamil, efflux pumps inhibitors, and he saw some enhancement of APDT efficiency in Candida, but that was with some super expressing recombinant strains. So I'm not pretty sure how that will be reproducible in a naturally strain that is producing more or different types of efflux pumps, which is the case of those Candida and Cryptococcus strains that we use. They're resistant to fluconazole. They have efflux pumps for that, and didn't change anything for us.
Okay. For discussion,
maybe it's also related to the concentration of the photosensitizer. I don't know.
Okay, this is important issue for your paper, the last paper. Okay, regarding clinical applications, I agree with Caetano on the use of topic use of PDT. And interesting, this is not restricted to human infection. Indeed, veterinary infection in pets or methicillin-resistant staphylococcus are very prevalent worldwide. In this case, antibiotics have little effect on pyoderma infection.
Pyoderma, sporotrix, sporotrichosis, leishmaniosis. There is not visceral.
Or fungal infection.
Yeah.
George, I agree with the plasmid copies and difference of inactivation of black KPC and blind. I don't remember of this point, but Caetano is true.
Yeah,
It's
I
an
agree.
explanation for the difference in your figure. And biofilm is important, and most likely the exposure to PDT should be different, and with high doses and sensitizer and exposure time.
Yeah, it is. We have done several experiments that the literature is very rich on biofilm inactivation by PDT as well, with some very beautiful images, 3D confocal microscopy, and so on. About the mechanisms, I don't know. Maybe the degradation of biofilms depends on other stuff, such as carbohydrate degradation for the extracellular matrix. But yet, to detach those things from the surface that it first docked, we need to degrade some proteins as well. And to actually kill the cell, we can't just spread the cells through a medium, through the bloodstream of a person with the infection. We actually want those cells to leave this colonized biofilm dead. We don't want them to be metabolically active. And I think that this death will be caused by the very same mechanisms that we show here.
Okay. Finally, cationic sensitizer are very interesting to be tested in the future. Very interesting, really. Well, okay. I invite the member for a private and brief meeting in order to perform the final decision. Please, I need that no member disconnect. Marta remains connected. Caetano, I send you a message for connect again. 2 minutes. Quick, okay?
Newton, maybe it's better we to open a new room because we have the transmission by
this YouTube. One is on YouTube. That's right, Marta.
Okay,
I can open a new room.
Okay, Caetano, could you send the link for us?
Marta is doing that.
Yes.
Marta, okay.
She's making a room.
You guys go there and I'll wait the invitation by email for marketing.
Thank you, Marta. Thank you so much. Okay.
See you soon, guys. Be kind to me. I think you guys should leave this room, otherwise we will also listen here.
So we're going to leave.
Yeah, you go to another room, so you can. That's how the procedure is made here. So you take the final decision if I deserve to be a doctor or not, in secrecy, and then you come back to this room to give the news.
Okay.
So Marta, Ma?
Yes, a moment, please.
Não tem mais
I'm looking for the email of Professor Jorge.
Usa o que eu mandei hoje de manhã.
Eu não recebi e-mail seu hoje de manhã, o Caetano. E eu estou procurando no que você mandou. Jorge, por favor, você está me ouvindo?
Ele já saiu. Eu vou pegar aqui o e-mail dele, já pego aqui.
Então fala para mim, Caetano, por favor.
Sampaio@usp.br.
Sampaio@usp.br. Eu mandei.
Beleza.
Claro que eu não mandei para mim, né? Hi, Mark. I'm here. Nice to see you. Everything is okay? Oh, Newton? Pronto, Paulinho.
So, what are the news?
Wait a moment, boy.
Ok. ¿Me escuchan? ¿Me escucha ahí?
Yes, Newton. We are listening.
On the 1st April 2021, the defense of the thesis of Mr. Caetano Sabino was made, which presented to obtain the PhD degree by the graduate program of the School of Pharmacy of the USP in Brazil, with the title of the work, Inativação, fotodinâmica, antimicrobiana, mediada por azul de metileno, Análises de cinética de inativação y mecanismos bioquímicos. And after the evaluation process by Professor Michael Hamblin, Mark Wainwright, and George Sampaio, the final decision is to approve. Caetano, congratulations to the new doctors. Caetano.
Well done, man. Dude.
Caetano, listen to me. Thank you for your friendship, firstly, and your participation in our research group, and the opportunity to work together in a topic that is very important for microbial growth resistance. Your work was performed in a perfect way. For us, was a new therapeutic alternative to be explored in research, and the results were very interesting, including for viral strains. We all know of your scientific capacity, and you have a promising future. I hope I can continue working with you and wish you the best in your new business. Thank you so much, Caetano.
Thank you so much. Thank you, Newton. It was a very important time for me working with you. I could extend so much of my knowledge, and also thank a lot for your friendship, your support, as well as for Martha, who has been withstanding me for a long time
Yes,
now.
I told them you are my oldest son.
Yeah. So we've been doing this thing for what now? 11 years. It's been an amazing time. It's been super productive. I'm very thankful and proud of this work with all of you guys, including Mike Hamlin, who took me in his lab. What, 9 years ago, 10 years ago, I don't know.
It's quite a while ago, Caetano.
I was a fresh man in university, first undergraduation in physics, and knew pretty much nothing. And you gave me a lot of trust and space to grow. It was amazing for that time. I stayed there for 4 months and came back with another level and capacity of my scientific contributions.
Yeah, as I remember, you spent a lot of time running around forming collaborations, but they all turned out to be worthwhile. I was quite impressed with that.
Yeah, it was really cool. We could engage collaborations with several people, even people who most of the folks there that I doubt that we could have a collaboration. We got it. And you know how hard I worked during that time. My thing was pretty much try to work longer and produce more than everyone else, because I had such a little time to spend there for what I could get. So it was really amazing. And like in 4 months, we had 4 papers. It was really nice. So thank you very much, Mark. When I knew Mark, the super dude, I still remember first time that I met you. I was requesting you a preprint of a paper, phenothiazines against melanoma, and the online version that we had, the graphs were all scrambled up. That was very bad figure quality. So I asked you that, and you brought me the whole preprint, like beautiful signed, autographed by you. I still have that in the very same
You sent to
folder.
me.
Yeah, I still keep that in the same folder where I keep all the awards and everything because I feel like the same thing there.
Oh, wow.
As well as the recommendation letter that Mike wrote to me a few times. I also keep in the same thing because I feel they're as much or even more valuable than any award that I ever got. So thank you very much. Thank you all for the friendship, for the trust, for the work together. Thank you very much, Professor George. We just met today and we never had a closer relation before, but I hope that from now on we can collaborate and cooperate, work together, and bring some new studies to our area and try to and punch off those drug-resistant bugs that we don't like. Thank you all very much.
Okay, this session is ending, and thank you so much. Thank you so much, and be careful.
Good
Goodbye.
to see you. Good to see you, Mike. Good to see you, Mike.
Bye, Martha.
Bye-bye. Newton, I would like to talk with you a moment, please.
Okay.
Okay.
Bye-bye. Pode ficar também.